Thank you, James, and thank you for inviting me. Yeah, you might see where I'm going here. I'm sure all of you have seen this slide. Oh, okay, that advances it. 10,000 years ago, the human species went through a major shift in its food supply. When we changed from a hunter-gatherer species, which we had been from whenever the first humans arrived, however they did. That's all up for speculation. But that shift to planting crops and raising animals from hunter-gatherer to agriculture happened roughly 10,000 years ago, earlier in some places, later in others. Anthropologists have been able to, by studying the skeletons from that era, on which side of the divide they were, and as we progressed through 10,000 years of agriculture, it's clear that we paid a price for that shift. We became smaller, weaker, more degenerative diseases, less robust in our health, but it allowed us to develop civilization. Now, the people that had the idea, hey, let's plant these seeds and stay here, they had no idea that they were the foundation of civilization. They were, just doing stuff, and it caught on. And, perhaps there is a collective consciousness that we share and a collective unconscious that we share. So, since the Industrial Revolution, we have been undergoing an increasingly rapid shift in our food supply that I submit is more drastic than the one our ancestors went through 10,000 years ago. First, we started using animals to pull plows, and then we invented a steam engine, and then we invented engines. So we gradually industrialized the process of producing food. In post-World War II, we started using NPK fertilizer with leftover explosives. Turned out it made great fertilizer. But after 50 years of only nitrogen, potassium, phosphorus going into the soil, we've depleted magnesium and chromium and zinc and lots of other trace minerals, and we've hybridized the food, and then we've genetically modified it. On top of that fundamental shift in the staples of the plants and animals that make up the food system, and let's not forget factory farming and all the antibiotics in the animals. So huge, huge shifts. It doesn't, I had to just step back and say, wow, this is, if you look at this in the context of the last 10,000 years, this is a huge shift. And particularly since World War II, then the rise of the food processing industry. And its goal was to make cooking obsolete. And they almost succeeded. They focused on maximizing shelf life, eye appeal, taste, and convenience. Nutrients were not on the table. They were not part of that equation at all. And at the same time that was going on, there's been a rise of the nutritional supplement industry. And I submit that that was a collective response, intuitive response to, hey, our food supplies really being depleted. We need to be able to capture and concentrate and stabilize nutrient factors to put back into our food supply. So I see it as kind of like a yin and a yang. And at the same time that we've dramatically altered the food supply, we've dramatically altered the environment. These so-called hallmarks of cancer were developed over the last decade in a couple of different steps by scientists at MIT. And they are qualities that we see in cancer cells. And one of the things I think that has really slowed down progress in cancer research is the focus on the cancer cell, and now the focus on the DNA of the cancer cell. So there's a lot more going on than just these changes that we've found in the nucleus of the cell over 100 years ago when scientists started noticing the chromosomes looked funny. And the ability to understand what's going on with the genetics is fundamental and very important, but as the title says there, it's not the whole story. So did you know that if you take the nucleus of a cancer cell and transplant it into a healthy cell, so you take out the nucleus of the healthy cell, insert the nucleus of the cancer cell, it becomes a healthy cell. All those DNA mutations and things, they don't drive that cell into cancer. But the vice versa, transplant a normal nucleus into a malignant cytoplasm, it remains a cancer cell, and the nucleus acquires all of these new DNA damages. It's a little bit of a chicken and egg issue of which comes first, but clearly there's a great deal going on in the cytoplasm, and things like reprogramming cellular metabolism. We know from Warburg's time that oxidative glycolysis becomes the main source of energy in cancer cells. And here's an example of mechanisms that, these are just examples, there's dozens that can enable each one of these. But if you inactivate the p53 gene, you develop the ability to evade growth suppressors. And people who inherit a defective p53 gene have Lee-Fraumeni syndrome, and they get lots of cancers from early in life, because the p53 gene is very important in allowing cells to repair themselves when they've been damaged. So if you get a lot of TGF-beta, you can avoid immune destruction. Telomerase gives you replicative immortality. Redirecting inflammation-promoting cells by bringing macrophages into the tumors, which tumors do, and getting them to make inflammatory mediators for them. Gives you the inflammation piece, many other aspects to that. If you inactivate one protein called E-cadherin, it unmoors the cancer cell and then it can travel. So activation of invasion and metastasis. Angiogenesis is growing a blood supply. VEGF is one of 20-some angiogenesis growth factors that we know of. If you inactivate DNA repair genes, you get genome instability and mutation, another of the hallmarks. And high levels of insulin-like growth factor, which you get from a high refined carbohydrate diet, helps enable the hallmark of resisting cell death, aerobic glycolysis, deregulating cellular energetics, and activating cellular oncogenes. So that's just a quick trip through what we know about the somatic mutation hypothesis, which has been the driving force of cancer. Now, I'm really, really pleased to see that the microenvironment, studying the microenvironment of the tumor is actually happening in academic research institutions. And that microenvironment is the main thing that we can affect with diet and lifestyle and functional medicine. Here is a graph of cancer incidence and cancer mortality. And these little blips here, that's when PSA screening started for men and got popular, and mammograms for women. So there was a spike of diagnosis of early disease, and a lot of that early disease was probably not clinically relevant, probably never would have become cancers. But you see that over 25 years, that is going up, and the mortality is going down. It was just announced that in the last 25 years, this goes to 2009, in the last 25 years, we've had actually a 25% drop in the overall mortality of cancer. So what does this mean? We're getting better at treating it. We're getting better at finding it earlier. It's a little controversial how much that plays. You've all seen this slide. That's what we're doing. We're getting better at treating it, but nobody's turning off the faucet. So the problem in cancer is that there's not one faucet. There's maybe 1,000 faucets. So some of those big faucets are the things that the functional medicine community can really help. And that's diet, lifestyle, testing of terrain markers and optimizing them, and going through the whole functional medicine matrix. But there are some of those faucets, like environmental chemicals, environmental pollution, that we can't turn off. And the trip to turning those faucets off is a very gnarly mix of money and power and politics and belief systems, and hopefully our future generations will be able to figure that out. This is just a quick trip of some of the things that you can do with basic functional medicine in primary and secondary cancer prevention. So primary prevention means preventing cancer from occurring in the first place. Secondary prevention is somebody's been diagnosed with cancer, they've been treated for cancer, and now they're hoping not to relapse. So that's secondary prevention. And I really encourage doctors to not be intimidated about working with cancer patients. They need you as much as they need an oncologist, in some cases more. So find and fix leaky gut. Everybody that's been treated has it. SIBO is also common. Try to get people off of PPIs and optimize digestion and elimination. Optimize their redox status. One point about the whole argument and concern about antioxidants interfering with cancer treatment is that scientists who don't know botanical medicine and nutritional medicine in a deep way generalize. So they do an experiment with rodents with N-acetylcysteine and vitamin E, and they show that it interferes with chemotherapy treatment. And they say antioxidants interfere because N-acetylcysteine and vitamin E are antioxidants. However, the huge range of antioxidants in botanical medicine involve mostly the class called polyphenols. And what we're learning is that polyphenolic antioxidants act as oxidants in the cancer cell, and they act as antioxidants in the normal cell. So they change what they're doing in response to the environment that they find themselves in. So beware of those sorts of generalizations by scientists who just don't know enough. So assess environmental exposures and assist detoxification. We're now beginning to talk about the exposome. We have the genome and the proteome and the transcriptome, and we have the exposome, which is the history of all of our exposures, and we're learning to be able to measure that. Optimize the functional medicine matrix. Optimize diet, lifestyle, emotional health, social connectedness. I've co-authored a book called After Cancer Care that covers all of that, so you can give that to your patients as a primer, both for primary and secondary prevention. So now I want to talk about another piece, a piece that we have a lot less control over. We do have a section in After Cancer Care about the things you can do to reduce your exposure to carcinogens and chemicals in general. And what it's starting to look like is that chemicals that have been tested and been shown not to be carcinogens, that is, they're not complete carcinogens, when all together, 5 or 6 of them can become a complete carcinogen. And they've never been tested together. I've been thinking about this for 30 years, and finally somebody is looking into it. And a man named Leroy Lowe was the founding voice and energy in the Halifax Project, a collaboration of nearly 200 cancer biologists and toxicologists. This was published a year and a half ago in Carcinogenesis. And I'll just read that slide because it's so elegantly stated. Historically, the axiom, the dose makes the poison, has had some merit. So many people remain skeptical about the idea that adverse outcomes can result from minute exposures to commonly encountered chemicals. But we are now at a point in time where our knowledge of the biology of cancer has advanced considerably. And we know that carcinogenesis can begin when key events have occurred in a single cell, between cells, or in the surrounding microenvironment. So the idea that low-dose exposures from many environmental chemicals acting together might serve to instigate, support, or fully enable carcinogenesis no longer appears to be an unreasonable assertion. I'd encourage you to just, Google the Halifax Project and keep an eye on it. It's leading the way towards potentially very important discoveries about how we can protect ourselves from the things that go beyond our diet and our lifestyle. Now, diet and nutrition are incredibly important because we know from many animal studies that deficiency of single nutrients can dramatically impact the ability of the organism to detoxify chemicals. We know that a rodent pregnancy that's deficient in zinc results in 3 generations of immune deficiency, even though they're fed zinc at adequate levels. And it takes 3 generations for it to come back. So what I wanted to do is just a very quick run through. There's more detail than I have time to go into, but just to give you a flavor of what's out there in terms of plant compounds and plant extracts that have the potential to block the effect of these groups of partial carcinogens on enabling the specific hallmarks. So I think we're at the threshold of being able to design scientifically based chemoprevention models. Chemoprevention is the name that was given to preventing cancer by taking something. So they tried, let's try beta-carotene, let's try vitamin C. But none of that was based on any very sophisticated science. So these are the, you can look at redox balance by oxidized reduced glutathione, liver detoxification path 1 and 2, optimize those, optimize methylation status, anti-inflammatory agents, methylating agents. This is all modifying gene expression. All of these plants are active against VEGF and against about a dozen other angiogenesis promoting compounds, but they're very weak effects, where Avastin is a very strong effect against only one angiogenesis growth factor. So the epidermal growth factor receptor can be restored or protected by green tea, grapeseed extract, etc. The p53 gene is supported and can be normalized with things from that group. And you see the cruciferous vegetables, long known to have a role in cancer prevention. HER2/neu, we have a lot of women with breast cancer and both sexes with esophageal cancer that's HER2/neu positive. So along with targeting HER2 with antibodies and pharmaceuticals, DHA from fish oil, olive oil, flaxseed, genistein from soy, quercetin, and emodin are all enhance that activity and can prevent development of resistance. Inflammation, we know, is fundamental to the development of cancer. We can go through and test the immune system, as Jeff Bland was talking about, looking at things like neutrophil to lymphocyte ratio. I like that to be 2 to 1. So that's a goal. It's something that their oncologist isn't looking at. So you can use things to support the lymphocytes and normalize T-cell subsets. Working against immune evasion, you've got a whole laundry list of things. So if we can, this is inducing cell cycle arrest, which you want to do after treatment's done to maximally inhibit any residual cells from growing. It's pretty clear that most people that have had a significant cancer and have a long go into remission and are for all intents and purposes cured still have cancer cells in their body in a dormant state. And they can be activated by things like car accidents or great periods of stress, things like that. So that's something to keep in mind. Not particularly something you want to share a lot with patients because it tends to induce anxiety. Oh my God, I've got cancer cells sitting around. And if they are, you just say, well, use that to be sure you get your exercise and your stress management practices. And I tell people use cancer as a lifestyle coach. It's the best lifestyle coach that there is. It doesn't take excuses. There's no, my dog ate my homework. You do the work. And you can induce redifferentiation. That's not a hallmark, but probably should be because dedifferentiation is present in virtually every type of cancer. And the more dedifferentiated they get, the more embryonic-like they get, the more aggressive they are. Inducing apoptosis, that's programmed cell death. And there's a lot of you see the same guys showing up because they're multifunctional. Inhibit invasion and metastasis. Lots of things at our disposal. Supporting anti-angiogenesis, lots of things at our disposal. William Lee has a very good TED talk about eating for angiogenesis. Not going to go into copper chelation. I just wanted you to get a flavor of that, the sort of the palette that we have to paint from. And as we develop the science of studying the effect of the exposome on us, I think it will become very possible to generate specific prevention programs that address the exposome of the environment, which is probably a very significant chunk of the cancer that we see. Most of the experts in the field say, with diet and lifestyle, we could probably prevent 50% of cancers. What's the other 50%? It's probably the environment. We've got 3,000 chemicals licensed to go into the food supply, 80,000 into the environment. With what we know so far from the Halifax project, there's no way that that's not a very significant burden. And so if we can generate the data about which groups of chemicals are particularly high risk, and by looking at the exposome in the patient, we can then generate specific protective combinations of plant medicines to protect us from that. So I think it's a very exciting time, and you in the functional medicine community have the opportunity to turn a lot of those faucets off from just poor diet and sedentary lifestyles and high stress. That's a devitalized diet, high stress, and toxic exposure is a recipe for a cancer epidemic. And we may be on the threshold of one. So I think it's important that this work move forward, and I have a lot of confidence in the adaptability of Homo sapiens to figure this out. So thanks very much.